Thank you. Hi, Michael Peluso. And we're happy to hear your talk.
Hi there. Let me just try to share. It is very, very early here in San Francisco, so I'm sorry if my brain is not functioning fully. I'm very sorry that I couldn't be there in person. I wish I could be. Um, I'm going to talk this afternoon, about targeting viral persistence in post-COVID syndromes. And what I decided to do is really provide an overview of where I think the field is at with regard to this. We're going to learn more about clinical trials that are going to report out over the next few months. So this is sort of a big picture step back of where we are and where I think we need to go. So my experience in this really comes from our program in San Francisco, which is called LINC, which stands for Long-Term Impact of Infection with Novel Coronavirus. And we've been studying long COVID for over 5 years now, really since the earliest days of the pandemic, following over 1,000 individuals, conducting thousands of study visits, and supporting many collaborations. We also have a tissue biopsy program that is really focused on identifying signs and mechanisms of viral persistence, an imaging program, a clinical trials program, and are beginning to expand our work beyond long COVID to other infection-associated conditions, including pre-pandemic ME/CFS. So as I'm sure you all know, there is evidence of pathogen persistence in many infection-associated chronic conditions. There's compelling data about enterovirus persistence in ME/CFS, in particular from John Chia's group down in LA. There is really interesting data about Ebola persistence in the post-Ebola syndrome, another condition that many in our group study. Obviously, remnants of Borrelia and post-Lyme syndromes. And across all of these syndromes, the role of herpes viruses, DNA viruses like EBV, is thought to be important. There's a lot of precedent for persistence of RNA viruses, and this is a really nice review written by Diane Griffin, who passed away last year, talking about both the viral factors and the host factors that can lead to persistence of these viruses, which are otherwise expected to be transient. So in the first section of this, I want to talk about where we are with viral persistence in long COVID, and these are the 3 points that I want to make. I think we know that SARS-CoV-2 persistence occurred in the first few years of the pandemic, when people had minimum pre-existing immunity and infections were more severe. We think that this is still occurring with modern infections, but honestly, the virus has evolved so quickly and the variants have emerged so quickly, and immunity has changed sort of beneath our feet, that the science is really still catching up with the evolving pandemic. And we don't know for sure that this is still happening. And what we don't really know is whether viral persistence is a cause of long COVID or a consequence of something else that also causes long COVID. So this is summarizing that first point, that SARS-CoV-2 persistence occurred at least early on in the pandemic. This is a really nice review written by Amy Proal, cataloging the dozens of studies of blood and tissue demonstrating SARS-CoV-2 persistence. Most of these studies were small, none of them was perfect, but there were similar findings across multiple laboratories, multiple orthogonal assays, demonstrating basically the same thing. And to me, that's really compelling, that there is something there. Importantly, almost all of these studies were pre-Omicron and at best early Omicron, so we're still waiting for more data to emerge in the Omicron era and especially in the modern era. When this first started to be reported, I basically said I'd believe it if I saw it in our own cohort here in San Francisco. And so in collaboration with David Walt, who has a single molecule array assay, basically an ultrasensitive ELISA for SARS-CoV-2 antigens, we studied pre-pandemic samples to establish the specificity of the assay, which was 2%, which was 98%, which is good but not perfect. And then when we studied post-pandemic samples up to 14 months post-COVID, we found a significantly higher prevalence of circulating antigen up to 14 months after the infection. This was twice as common among those who were hospitalized compared to those who were not hospitalized. And it was 3 times as common among non-hospitalized people who just said that they were really sick compared to people who said, oh, it was mild, my infection was mild. So as a clinician, to me, this really lends a lot of biological plausibility to the finding, and we've now extended this observation out beyond 24 months. This has to be coming from somewhere, and so I mentioned we have a tissue program in San Francisco where we perform gut biopsies. So what you're looking at here are gut biopsies taken by flexible sigmoidoscopy. The first set of slides are control samples collected before the pandemic, and you can see that there's no signal. We're looking for green for signal here. On the second column, you can see the post-COVID samples, where we think we're seeing SARS-CoV-2 spike encoding RNA. The top participant is 6 months post-COVID with long COVID. The bottom participant is 2 years post-COVID with long COVID. There's no known interim reinfection in either of these cases. And the signal is all located in the lamina propria, which is the sort of immune surveillance layer below the epithelium. It's not in the epithelium, and it's in areas that are dense with CD68 infiltrate, which suggests sort of myeloid macrophages. And actually, if we look, we can see double-stranded RNA, which suggests that the virus is actively moving through the life cycle. So I mentioned there's less certainty about whether viral persistence actually causes long COVID, and I just wanted to talk about these 2 papers that were discussed over the last year. So the first one is from the National Recover Program, where a postdoc, Zoe Swank and David Walt's lab, managed to tie the detection of circulating antigen up to a year post-COVID to the presence of long COVID symptoms. This was the first statistically significant relationship that emerged between detectable antigen and long COVID symptoms. And then we presented earlier this year at CROI an observation that we could actually link antigen detection with some, but not all long COVID symptoms. So we see a relationship, for example, with neurologic long COVID symptoms and cardiopulmonary long COVID symptoms, but not with more general symptoms like fatigue or GI symptoms. So now to shift to sort of what I think we need to really answer these questions. We need studies that are aimed at preventing viral persistence from happening in the first place. It would have been very nice to have had these studies 2 or 3 years ago. We're still trying to launch them. But this is really important to see whether this can be prevented. We also need more studies aimed at targeting viral persistence once it's already occurred, sort of reversing viral persistence. And we need to make sure that we're not making huge sweeping claims from a few imperfect studies, whether they're positive or negative. I really think that we need a dedicated biomarker development program to actually figure out how to measure this in a scalable way. Not everyone can get a gut biopsy. And with that comes the need to admit the limitations of our current biomarkers and to stop saying that we don't need biomarkers. And then I think we also need to keep an open mind about whether or not this is ultimately going to be the right target. So just a few points I want to make with regard to those items. So a really important question is how acute infection might influence viral persistence. These are data from our acute COVID study, where we were able to get daily nasal PCR samples for the first 3 weeks post-COVID in individuals with a new SARS-CoV-2 infection, and then relate the maximum viral load in the first couple of weeks to people's clinical status 3 to 6 months later. And what we found was that people who were destined to go on and develop long COVID had a 2 log higher maximum viral load during the first couple of weeks of infection. And so that bears a question: do acute events really determine who's going to go on to develop long COVID, and is this driven by a mechanism like viral persistence? There was also data presented this year at CROI that showed that people who have viral rebound, whether or not they're symptomatic, have a 13-fold increased odds of developing long COVID. And one wonders whether that is related to virus sticking around. So with regard to that, we really need to know whether we can target the virus early and aggressively upfront to prevent the downstream cascade that ultimately results in long COVID, and whether that interferes with mechanisms like tissue damage, viral persistence, or maybe other mechanisms that we're not even talking about, and whether that can affect long COVID. If we can establish proof of principle for long COVID, this would be transformative, I think, for preventing other infection-associated conditions. And there are very, very few studies, and none in the US, that are doing this right now. The next question is, does targeting viral persistence matter? This is a schematic from a review paper a group of us recently wrote, talking about all of the different forms in which the virus might persist. I don't have time to kind of go into all of the detail here, but this is important because based upon the form in which viral persistence might be occurring, you might choose a different therapeutic modality to target that persistence. And that can range from sort of improving immune system function to directly targeting the virus with antivirals. There were a lot of case reports over the last couple of years suggesting that there could be promise with these strategies. These are all anecdotes of people who were with long COVID, who took Paxlovid and got better, or received a monoclonal antibody and got better. In some cases, these were sort of miraculous returns to health, but all sort of anecdotes, not controlled studies. The controlled studies that have come out so far have not supported the findings of those anecdotes, unfortunately. So there are two reasonably sized studies of antivirals in long COVID. The Stop-PASC study at Stanford, the PAX-LC study at Yale, which unfortunately showed no overall benefit between treatment and placebo in people with long COVID. But there are a lot of caveats to these sort of first-generation studies. They couldn't confirm ahead of time that the participants had viral persistence. The tissue penetration of drugs like Paxlovid may be too limited to get to the places where it needs to be. 2 weeks of treatment might be far too short. In animal models of coronavirus persistence, it requires 8 to 12 weeks of treatment to eradicate the virus. Unfortunately, there's no toxicology data available to support such long courses of treatment in people. And we don't have biomarker results from these studies yet. I want to mention that Recover Vital, which is a 900-person study of 2 to 3 weeks of Paxlovid versus placebo in long COVID, is going to report out in the next few months, and I think is going to be hugely informative. But even if that study is overall negative, I think that there's a lot of work that still needs to be done. We are in the final stages of wrapping up the analysis for our study of a monoclonal antibody in long COVID. This was a proof of concept randomized trial of a monoclonal made by Arium Therapeutics in people with established long COVID, single infusion, long half-life. We followed people for up to a year afterwards and basically measured everything to try to do a deep dive into whether we could detect differences in symptomatology and different biomarkers over time. Importantly, in this study, half of the participants did pre and post gut biopsies, and so we'll have the ability to really understand what's going on in the tissue compartments as well. So I just want to emphasize that in these early long COVID trials, the top line results don't always tell the whole story. And so even when these studies have been positive, I think they're not super clear. So this is a study that you may be familiar with of a symbiotic preparation that showed a significant reduction in long COVID symptoms. But if you look sort of under the hood, there wasn't really any improvement in quality of life in the long COVID group compared to the placebo group in this study. So even though there was a benefit, it's unclear what the impact of that benefit on the patients was. And then this was a study that was negative, a really well done study of an RNase molecule, which basically can degrade cell-free RNA. that was overall a negative study. But again, if you look under the hood of this study, what you see is that there was a pretty impressive response just in the women in this study and no response in the men. And so, as this first round of studies in long COVID reports out, I think that we need to be really thoughtful about how we're interpreting the results. And then the last point I want to make is that we really need better biomarkers. So I spend a lot of time knocking on the doors of pharmaceutical companies trying to get access to drugs. And what they say over and over is we want to help, but we need a biomarker first. At the same time, as we were trying to launch this first round of studies, many stakeholders, including patient advocates, were emphasizing that we really didn't want to limit who can enroll in the studies to people who had a certain biomarker profile. And so it's sort of a catch-22 here. These tissue-based markers that I talked about are not really available and are not really accessible. We need biomarkers that can be scaled. There are a lot of issues, not necessarily with the specificity of these assays, but with the sensitivity, and in particular, with variability within a person over time, which makes them really hard to apply in clinical practice or in screening for a clinical trial. And so what I really think we need is to take a big step back and really invest in this. We need a large multi-institution dedicated biomarker program that is focused on biomarkers of viral persistence or other biomarkers of long COVID. And we need to admit that we're not really going to be able to accelerate the field in the way we need without doing this. This is a schematic of what the current problem is, which is basically that every individual lab is developing its own biomarker, and there is no head-to-head comparison between these markers. And so what we really need is a central cohort with deep phenotyping, repeat measurements, abundant biospecimens that can be distributed in a blinded manner to the laboratories to all run their assays on the same specimens from the same people, with central data analysis to see what performs best.
So we need to
My last slide is that I really think that this is an important model for other infection-associated chronic conditions. And so what we learn from developing this in long COVID, I envision as being able to be applied to other conditions, including ME/CFS, to accelerate progress across all of these conditions. I'll stop there. Thanks so much.
Thank you so much. We're running out of time, but on the internet there were a lot of questions. Is the SARS-CoV-2 reservoir the nicotinergic receptor? That was a question which comes here from several patients. When giving that, when you use nicotinergic patches, you get better clearance of SARS. Is that any evidence for this?
Yeah, I think that that's really unknown. I mean, I have heard some of those stories about people having improvement. I've also, seen our own participants who have tried it and not really had any improvement. I'm not aware of any trial-based data to support that.
Yeah, thank you very much for the presentation and the short comment on this question. Yeah, we need more data. This is quite clear. Thank you very much. And we move